Hi dear students, I am Dr. Mashur. Welcome to my class. Today I would like to discuss introduction to DNA replication and different models of replication and Meselson and Stahl experiment to prove DNA replication is a semi-conservative model of replication. Now let's see DNA replication. DNA replication is the process by which the genetic material DNA is copied. Okay, DNA replication is the process by which the genetic material DNA is copied. And in this DNA replication, the original strands are used to synthesize template for the synthesis of new strands. Okay, the original strands that are used as a template to synthesize new strands, and this replication process is occurred in the life of a cell at appropriate time and is occur very quickly and very accurately. Okay, is occur very quickly and accurately during the appropriate time of a cell. Okay, normally this DNA replication is occur during the time of cell division. Okay, and this DNA replication process is relies the complementarity of DNA strands. Okay, DNA replication is relies the complementarity of DNA strands. That means if a DNA strand having one DNA template strand having a nucleotide, then this complementary strand same position will be having T. That means it follows a TGC rule, and that means it follows Chergaff's equivalent rules. Okay, and you can see simply explain this DNA replication as DNA replication is the copying of genetic material DNA, and during this process, two complementary strands of DNA strands come apart, and each parent strand is served as template for synthesizing new strands. Okay, each this parent strands come apart during the process of replication, and each strand serves as a template for synthesizing new strands. And this newly synthesized strand that is known as dot strand, and the other strand that is known as parent strand. Okay, DNA replication is the process by which DNA molecule is copied, genetic material DNA molecule is copied, and the original DNA strand is served as a template for synthesizing new DNA strand, and this process occurs very quickly and accurately at appropriate time during the cell life. And DNA replication relies the complementarity of DNA strand, that means it's follow and a TG0, that means Chergaff's equivalence rule. And during this process, the two DNA strands are come apart. Okay, two complementary DNA strands are come apart, and it will serve as a template for synthesizing new strands. Okay, these two other strands will come apart and then serve as a serve for synthesizing, serve as a template for synthesizing new strands. Okay, and the two newly synthesized DNA strands that is known as dot strand, and the two original strands that are known as parent strand. In this figure, you can see this parent strand, and another one is that parent strand is violet, and that form under for replication is formed to dot strand, that is one is blue, and two blue strand is formed, and the parent strand also is the color in the violet. This is about the introduction of DNA replication. Now we can see some detail about the DNA replication. Okay. In 1950s, there are different mechanisms were proposed for the replication of DNA. Okay, 1950s, there are different mechanisms, different models of replications were proposed in 1950s, in late 1950s. And the first model is a conservative model, second one is semi-conservative model, and third one is a dispersive model. Okay, first one is a conservative model, second one is semi-conservative, and third one is a dispersive model. So we can see each model separately. In the conservative model, after replication, both parent strands stay together after replication. Okay, in this conservative model of replication, both parent strands will stay together, and this daughter strand also stay together. Okay, this is a conservative model of replication. In this figure, you can see one black parent strand is the black two parent strand is there, and from the other end go for replication, and it forms as two double stranded DNA, and one double stranded DNA having two original parallel strands, and another double stranded DNA having two strands of new daughter strand. This is known as a conservative mode of replication, and in the next one the semi-conservative mode of replication. In semi-conservative mode of replication, after the double stranded DNA contain one is parallel strand and another one is strand is a daughter strand. Okay, after replication, one strand will be parallel and another strand will be daughter strand. That is called as a semi-conservative mode of replication. And third, also in this figure you can see the semi-conservative mode of replication, that black color two parallel strand is undergo for replication and is formed replicated, and is formed one double stranded DNA with one black and another strand with blue, another on second double stranded DNA having one black and one is new. That means one parallel and one new strand is there. In third one, the dispersive mode of replication, both strand having, that means both parallel and daughter DNA are interspersed in both strands following replication. Okay, after replication, this both parallel and daughter DNA is interspersed in both strands after the following replication. Okay, this is a dispersing model. Okay, first is a conservative model of replication. Okay, conservative model of replication. Conservative model of replication, both parallel strands stay together after replication.
And in semi-conservative mode of replication,
both this double strand DNA contain one parallel and one daughter strand. Okay, one parallel, one daughter strand. And in the case of dispersing mode of replication, this figure you can see dispersing mode of replication, both parental and daughter strand DNA are interspersed in both strand following replication. These are different models of replication. We have seen three different models of DNA replication. Okay, in 1950s, many scientists were worked to find out which model of replication is correct, and to prove that, Meselson and Stahl, two scientists, they were devised a method in 1958 to find out which model of replication is correct. Okay, in 1958, Meselson and Stahl, they were devised a method to prove which model of replication is correct. For that, they first grew bacteria for a several generation in a medium containing only N15. Okay, they were cultured the bacterium E. coli for several generations in a medium containing only N15. Okay, N15. Thus, N15 means heavy nitrogen. Okay, heavy nitrogen. And after that, and this cultured bacteria, we can they will call it zero generation, because is just before switch this bacteria into N14. Okay? Just before switching this bacteria into N14 medium, that's why there is known as zero generation bacteria. Okay, from this zero generation bacteria, they were isolated DNA and they were done analytical centrifugation. They were done analytical centrifugation this DNA, and in that analytical centrifugation, cesium chloride centrifugation, they were found that they were found that it's only produced one band, that was heavy band. Okay, it's produced. This means N15 medium containing bacteria, that means zero generation bacteria, produced one heavy band in cesium chloride centrifugation. Okay, and after that, from this zero generation bacterial culture, they were taken samples, a portion of samples from this zero generation bacterial culture, N15 medium culture. They were taken a portion from that and they were transferred into a medium that only contain N14. Okay, that only contain N14. Okay, that only contain N14. Okay, after first generation. Okay, that means after first generation. Okay, after first generation in N14 medium. That means after first generation means after one replication. After one replication in N14 medium, there were isolated DNA from this bacteria. Okay, they were isolated DNA from this bacteria and go for cesium chloride centrifugation. Okay, they will go for cesium chloride centrifugation, and that they were found that there is only one band is appeared, but it's not similar that of band formed in the zero generation bacteria. Okay, this first generation band that was lighter than the heavy band. Okay, lighter than the heavy band, but it's not much lighter than the light band. Okay, that's why they call this zero first generation band that was middle of this heavy band and light band. That's why they call this a middle band or intermediate band. Okay, first generation bacteria they given intermediate or middle band. Okay, this was the first generation bacteria experiment, and they allow this first generation bacteria to proceed to second generation. Okay, they allow this bacteria to proceed to second generation. In after the second generation, that means that bacteria undergo for two replication in N14 medium. Okay, they proceed to N14 medium, two replication. They completed two replication in 14 medium. That's why it's called as a second generation. And after second replication cycle, they were isolated DNA from that. Okay, they were isolated DNA from that, and they undergo for cesium chloride centrifugation. They undergo for cesium chloride centrifugation. In that cesium chloride centrifugation, they found two band. Okay, they found two band. One band is same as like it is seen in the first generation, like that. That means they found one intermediate band that is same which is appeared in the first generation, and in this case also second generation also heavy band was absent. Heavy band was also absent in second generation also. Apart from this middle band that is appearing in second generation and first generation, in second generation another one band is appeared, but that band is lighter than the middle band, that is light band. Okay, that is lighter than the middle band, that means it's a light band. Okay, that means second generation contain two bands, one is middle band and one light band. Okay, this is the second generation banding pattern. And after that, they allow the second generation culture go for third and fourth generation. Okay, after third generation, they were purified DNA and they were done cesium chloride centrifugation. In that, they have found that the light band become predominant. Okay, light band become predominant, but the intermediate band become faded. Okay, the intermediate band become faded, but the light band become more predominant, and the light band become as the generation is progressed, this predominance of this light band is more and more increased, but the this intermittent bands is become fade. This was the result they observed. I will explain again this method. This experiment. They first this Meselson and Stahl. They first grew the bacteria E. coli cells in N14 medium, N15 medium only for several generations, and this culture we can call as zero generation because it's just before switching this bacteria into N14. So this is known as zero generation bacteria, and they cultured this bacteria in N15 medium containing only N15 medium, and they were isolated DNA from this. Okay, they were isolated DNA from this, and they purified DNA, and they undergo analytical centrifugation. That means Cesium chloride centrifugation. Then the Cesium chloride centrifugation. In that, they were found that there only one band is appeared, that was heavy band. Okay, in zero generation bacteria, the appeared band was heavy band. Heavy band they were appeared, and after that, from this N15 medium, they were taken a portion of bacteria. Okay, portion of culture they were taken, and they were transferred into medium containing only N14. Okay, that medium containing only 140. Only the medium containing only N14. Okay, medium containing only N14. In that, this bacteria are allowed to grow one generation. Okay, allowed to go for one generation. That's why it's called a first generation bacteria. This made the bacteria grown in N14 medium for one generation. That is called as a first generation. That means this bacteria undergo for one cycle of replication in 14. Okay, after one replication, one generation, they were taken DNA from this and undergo for Cesium chloride centrifugation. Undergo for Cesium chloride centrifugation. In that centrifugation, they found that there is one band is appeared, only one band is appeared, but it's not similar to the band formed in the zero generation. This band was lighter than the heavy band which appeared in the zero generation, but this is not at the position of the light band. Okay, but it's in between the heavy and light band. That is called as an intermediate band. Okay, this is not the position of light band, not in the position of light band that we expected for the position of light band, but it is the position of intermediate band. Okay, intermediate band or middle band. This is the first generation band was middle band, and after that they allow this first generation bacteria to grow for second generation. Okay, after second generation, second generation means that bacteria undergo two cycle of replication in N14 medium. Okay, two cycle of replication in N14 medium, and they were purified in DNA, and they examined in the Cesium chloride centrifugation. In that time, they were formed two bands. One is similar to that is appearing in the first generation, that means the one is a middle band, and one another additional band is appeared, that is a light band. Okay, this is lighter than the middle band, so it's called as a light band. Light band is appeared in the second generation. One is the middle band, second one is the light band. Two bands were observed, but in the first generation only one band was used, that was middle band, and they allowed the second generation bacteria, they were allowed to go for third and fourth generation. Okay, in after third generation they were isolated DNA. In that they found that the light band, the lightweight band become predominant. The lightweight band become predominant, but the middle band become more faded. Okay, middle band become more faded after the progress of the generation. Okay, during the progress of generation, this light bands and this middle band become more faded. This was the experiment they conducted. This was the result they got from this experiment. From this, they said that semi-conservative mode of replication is correct. Okay, do you know how they told semi-conservative mode of replication is correct? So let's see. The starting culture, that is zero generation culture, they taken bacteria and they isolated DNA. They get only one band as well as heavy band. Okay. Then they told that this heavy band is because of the two strand of the DNA is only contain N15. That means heavy nitrogen. That's why it's given as a what? Heavy band. Okay. heavy band is heavy band because of that two strand only contain N15, that's why it is given N50. This band given as a heavy band. And in the first generation, they got an intermediate band. They got intermediate band. From this result, they told that this result shows that DNA replication is not a conservative model. Okay, DNA replication is not a conservative model. It may be semi-conservative or dispersive. Okay, DNA replication will be semi-conservative or dispersive model, but it is not a conservative model. How they told that it is not a conservative model? Because if it is conservative, if it is a conservative, there will be one parallel strand with the parallel strand will be with the N50 and the dot strand will be with the N40. Because in conservative model of replication, that parallel strand and dot strand will be stays separated. That's why we can see here, if it is a conservative model in this tube that I have to show two bands, one will be light band and another one is a heavy band. But this tube only show only one band, that was intermediate band. That's why they told DNA replication is not a conservative model. And next step, the second generation result, they told that DNA replication, that come from that DNA replication is
semi-conservative.
because if DNA replication was dispersive model, that time we won't get a pure light band. Okay, we won't get a pure light band because in the the second generation, there's two bands formed. One is intermediate band. Okay, intermediate band. They explain that in that intermediate band, one strand will be N14 and one strand will be N15. That way, that DNA is formed as an intermediate band. Okay, and another set of one band also is there. That is a light band. Light band that contains two strands with only N14. Okay, in this figure you can see that. Okay, in this figure you can see that one strand will be this one double strand DNA having N14 and N15 strand. That's why that forms an intermediate band, and another one band that's shown as a light band is because of is formed with N14. Okay, N14. The two strands are composed of N14. That's why is given light band. If it is a dispersive, this both strand will contain both N15 and N14. That's why it does not give a pure light band. That's why they told that this is not a DNA replication is not a dispersive model. Okay? That is, dispersive model does not never give a pure light band. Okay, it does never give a pure light band. That's why they told that this DNA replication is
Semi-conservative.
in third and fourth generation, why the intermediate band become morphoid and the light band become predominant? Because in the third and fourth generation, the hybrid band too become progressively faded because this intermediate band represent only small fraction of total DNA. Only represent small fraction of total DNA in this. Okay, that's why it's only give faded band. In first generation, you can see 100% intermediate band. But in the case of second generation, you can see 50%. This 50% intermediate band. And the third generation, you can only see 25% intermediate band. When it's go to fourth generation, that time only contain 12.5% intermediate band. That's why, as when the DNA replication is progress, that time the intermediate band intensity will be get faded. And this is from this method they have proved that DNA replication is semi-conservative. DNA replication is semi-conservative. I hope you understood this class. Thank you for listening.